In our last video on neurotransmitter release, we learned that when an action potential comes down to the axon terminal, it causes the inside of the terminal to depolarize. The depolarization of the terminal opens a new type of voltage-gated channel called a calcium channel, allowing calcium to enter into the axon terminal. It's the influx of the calcium into the terminal that causes these synaptic vesicles that are docked with the plasma membrane of the axon terminal to fuse with the plasma membrane, producing a pore whereby the neurotransmitter substance can actually be released into the synaptic cleft. And we talked about how it would cross the cleft and end up coming into contact with a receptor protein, and that by interacting with that receptor protein, it could cause changes in the membrane potential of the postsynaptic neuron. And in this video, we're going to take a look at the different types of receptor proteins and how neurotransmitters can affect those proteins, and what then the overall effect on the neuron's membrane potential would be. Now, this could be a really long video, and so I'm going to divide it up into 4 different parts. And what we're looking at right now is part number 1. Our main objective here is to understand a little bit about receptor proteins and about the different kinds of receptor proteins and what neurotransmitters will do to them. Okay, so what we'll do at this point is we'll take a look. I've blown up this receptor protein over here, and we're going to take a look at how the neurotransmitter affects the receptor protein. This, of course, these 2 lines represent our lipid bilayer, and here is the receptor protein. Receptor proteins don't actually look like this. This is just a representation. Something that's important for us to note, though, is that there will be a spot, which I've represented as this indentation down here, where the neurotransmitter can actually physically interact with the receptor protein. And this particular location is sometimes referred to as the binding site, or it could be called the receptor site. So we'll use those names interchangeably. What we have then is that neurotransmitter, as it crosses the synaptic cleft, it can fit down inside of the binding site on the receptor protein and cause a change to occur in the receptor protein. Now, depending upon the type of receptor protein, there are only 2 things. Well, let me back up and say that a different way. There are actually 2 basic types of receptor proteins. So when the neurotransmitter binds with the receptor protein, it's going to produce slightly different effects depending upon which one of those 2 types that receptor protein was or is. So let's take a look at the 2 types. First type is known as an ionotropic receptor. And the second type is known as a metabotropic receptor. Okay, sorry for the poor handwriting. That's a B. So it's metabotropic. Let's mark that as a B right there. All right. Now, to understand how these receptor proteins work, it's useful just to examine their names. You can see here in this particular name that one of the words is ion. And so we know something is going to happen with ions. What's going to happen depends upon the meaning of this particular portion of the word. And this actually comes from a Greek word, and that Greek word is tropy. I don't speak Greek, but I'm pretty sure that's close to the pronunciation. This means turn or turning. So if we think about it a little bit, ionotropic refers to some sort of turning or change in the ion. And as we'll learn, what actually happens with an ionotropic receptor is that when the neurotransmitter binds to it, the receptor protein becomes a channel and an ion will flow through the receptor protein. So we'll just put that here. Ion moves through receptor protein. All right, that's an ionotropic receptor. Now, if we look down here at the metabotropic receptor, we can see that there's a part here, metabo, that looks a lot like the word metabolism. And again, we have the tropic for the turn. And so what we're referring to here with a metabotropic receptor is that when the neurotransmitter binds to the receptor protein, we're going to produce a change in the metabolism or the chemistry of the neuron. Now, it's different from an ionotropic receptor because the receptor protein does not open up and become a channel. Rather, something else is going to change that will lead to movement of ions, and we'll look at how that happens actually in the next video, or actually the third video on receptor proteins. Before we finish this video, we need just a little bit more vocabulary to help us with our discussion. And we're going to turn to biochemistry for just a moment to understand the vocabulary. In biochemistry, there's a term known as, and the term is ligand, or it's also pronounced ligand, L-I-G-A-N-D. And it refers to a molecule that will bind with or join with another molecule. Usually the ligand is smaller and the molecule that it's binding to is larger. Well, you can see that that's exactly what's happening here. The neurotransmitter is, compared to the receptor protein, a relatively small molecule, and it will bind with or join with the receptor protein. And so we refer to the neurotransmitter as a ligand. And sometimes this kind of receptor protein is called a ligand gated channel, or alternatively a transmitter gated channel. Now, these ligands don't stay bound permanently to the receptor protein. So the neurotransmitter only binds with that receptor protein for a brief moment, and then it detaches and goes back out into the synaptic cleft. Since it's bouncing around, it's possible that it could once again enter the receptor site or the binding site and produce its effect another time. And so later, in a later video, we'll look at how do you remove these neurotransmitters from the synaptic cleft, because as long as they're there, there's the possibility that they may repeatedly bind and unbind with the receptor protein, producing their effect over and over. In summary, what we've learned in this video is that when the neurotransmitter crosses the synaptic cleft, it's going to come in contact with one of two different types of receptor proteins. One being an ionotropic receptor protein, which will open up and allow an ion to pass directly through it. Or the second possibility is that we'll have it bind to a metabotropic receptor protein, and that will produce some sort of change in the chemistry of the neuron, which will eventually lead to a change in the flow of ions. Not a direct change in the flow, but an indirect change in the flow of the ions. So that's it for this particular video. In the next video, we'll examine ionotropic receptors in a lot more detail.